So students, you have learned the different aspects of the metabolism of glycogen, specifically the synthesis of the glycogen, that is the glycogenesis, and the degradation of glycogen, that is the glycogenolysis. But always keep in mind that metabolism is not all about synthesis and degradation. Even more important in metabolism is the regulation of these different activities. If, if there is disruption in the regulation, that is disruption in regulation of the synthesis or degradation of glycogen or any other such molecule, that will lead to all sorts of problems. So now let's we see how the two activities, that is the glycogenolysis and glycogenesis, is regulated in our body. As I have just told you that the regulation of glycogen metabolism involve both the regulation of the synthesis of glycogen as well as degradation of glycogen. First, let's we talk about the degradation, that is glycogenolysis, because this is the process in which you get the glucose through the or by the degradation of glycogen. This is a very cell signaling based event. I mean, whenever we are talking about the regulation of any pathway, it most of the time involve very intricate, very sophisticated cellular signaling pathways. So here the cellular signaling pathway, which is mostly responsible for the degradation or even synthesis of glycogen, is the G protein link pathway or GPCR based pathway. G protein receptors, you know that are one of the most diversified receptors, one of the most common receptors found in eukaryotic cells, and they are very much involved in more than 50% of the pathways which are or activities which are taking place in our body. So the hormones which are involved, we have already been talking about these hormones, specifically the glucagon hormone. It binds with the GPCR protein. For instance, we can see here, this is the GPCR, and the hormone binds over here. And after binding, the one of the most classical way of or also known as canonical way of the activation through of a pathway through GPCR based signaling is that it leads to the then activation of certain sort of enzymes and molecules like cyclic AMP is formed. This cyclic AMP then leads to the activation of another enzyme, let's say protein kinase. And then this protein kinase, because this protein kinase is composed of two subunits, and the release of one subunit from the other leads to the activation of a protein kinase, and which in turn then activate another enzyme, which in turn activate the phosphorylase, and the phosphorylase enzyme is actually involved in the degradation of glycogen. So the phosphorylase is again in two form. Unphosphorylated form is the inactive form, while the phosphorylated form is the active form. So what will happen that through protein kinase, it gets activated because of the process of phosphorylation. So once the phosphorylase enzyme is activated, then it will lead to the degradation of glycogen. So in this way, you can see how the reaction or pathway starts upstream, most upstream of the cell, that is from the cell membrane. It goes down till the cytoplasm, and you get the response. And sometimes even it goes even further down till the nucleus, but not in here this case. But you will see later on that many of the pathways also involve the gene regulation. Then cyclic AMP binding is also a very important path. I have already told you that's a very important aspect of the what we say the activation of GPCR based activities, because the cyclic AMP gets activated or is formed because of the activation of the GPCR, and this GPCR, when they give rise to cyclic AMP then leads to the activation of, as I have already told you, protein kinase, and the activation of protein kinase then leads to the phosphorylation of enzymes, which leads to the activation of many other downstream signaling molecules. Sometimes it happens that without, I mean, cell needs things much more readily, much more rapidly. Sometimes then it can even go through processes which doesn't involve so many of the phosphorylation events, like AMP, adenosine monophosphate. It also signals or stimulates the activation of the enzymes which are involved in the what we say the degradation process, the degradation of the glycogen. So there are key molecules like AMP, cyclic AMP, GTP. They I mean they are activated, especially the AMP. It also leads to the activation of the process of degradation of the glycogen. Similar to glycogen degradation is the glycogen synthesis. The enzyme which was involved in the glycogen degradation was phosphorylase, while the enzyme which is involved in the synthesis of glycogen is glycogen synthase. And this glycogen synthase is also in active and inactive form, but little bit difference is that it's so in the glycogen phosphorylase, the active form was phosphorylated one, which was phosphorylated by pKa, protein kinase. Here, the active form is actually dephosphorylated, but outcome is the same, that whether it's phosphorylated or unphosphorylated, the thing is that you should keep in mind what is actually the active form. Once it gets active, then this synthesis of the glycogen will prevail. Now, an other very important aspect of cellular signaling is the signaling cascades. I mean, amplification of the signaling. It's one of the hallmark of cellular signaling, especially the G protein linked signaling, that you start with one or two molecules which are very upstream. For instance, certain hormones, they activate a GPCR receptor that then activate many what we say cyclic AMPs. Cyclic AMP then activate protein kinases, many protein kinases then activated many enzymes, and those many enzymes then activate further many molecules, and the activation of those many molecules give an outcome in a very amplified way. And that is how this amplification of a signal takes place. And this amplification is very much needed, because you not only need a very correct response, you also need a response with a full strength, with full intensity, so as to cope the need of the body. So that's why cellular signaling in this way make it sure that not only the response is timely, not only the response is very precise, but also that strong is very potent.